drug discovery for big pharma. So most of the drugs that are made today, the targeted drugs, particularly the targeted drugs, which are coming out of the Human Genome Project, start with understanding the protein structure. And the better you understand the protein structure, the better you can make a drug. And it turns out that there is a promise, a significant promise, that proteins can be better crystallized in the microgravity environment than on Earth. And so we're now going to really test that out. And one of CASES's goals is to engage commercial interest. And so that would be big pharma. And so you'll see on the CASES website some announcements with big pharma interest in projects in space. The second area of interest we found was in looking at osteoporosis. Osteoporosis is a significant disease affecting postmenopausal women in particular, and aging women. And so it turns out in space, astronauts are significantly affected by bone loss when they come back from the microgravity environment. And so it appears to be a very good recapitulation of the human problem. And so we're now looking into potentially putting mouse models up in space that could be tested with drugs or wild type and transgenic mice, comparing proper controls for these mice to really study and potentially develop drugs for osteoporosis. A third is muscle wasting. Again, another problem that came from the astronauts in their post-trip evaluations. We found they lost muscle mass, and that exercise, alone wasn't sufficient to prevent that problem. It turns out that the muscle loss, again, appears to recapitulate some of the human capacities we see where there's muscle loss, the human diseases where we see muscle loss. And so again, there's a pharma interest in potentially developing drugs to target muscle wasting. And we're also looking at other problems such as immune loss, which is a problem, and the potential for stem cells to be grown better in space than on Earth. We've had a number of unsolicited proposals submitted, I think about 35 to date, and we're progressing to fund some of those. And we've also had our first solicited RFP, and that was in protein crystallography. And so I think we're progressing on a number of fronts, as well as material science. There's been an unsolicited proposal there that will likely be funded. So all in all, very exciting and a very diverse group of experiments. So thank you.
Okay. Well, let me start by saying this is our first flight opportunity, and myself and my whole team is thrilled to be part of it. We're actually looking at our experiments are called Micro 6, and we're interested in analyzing Candida albicans, which is a yeast, and how it responds to the microenvironment of microgravity. There are a few things that we're looking at. One is Candida albicans is a commensal organism. It's common, but it usually doesn't cause many problems. But it is also a human pathogen, an opportunistic human pathogen. So when our immune systems take a hit and aren't functioning as well, Candida albicans finds an opportunity to cause infection, and those infections can be things like thrush or fungal nails. So we think it's important in its own right because of that potential. Because Candida does have this infectious potential, the role of these experiments, really the data that we're trying to acquire, is dual in function. One is we really want to assess whether Candida albicans poses any health risk to the flight crew. And then we're also very interested in learning what we can about how Candida albicans responds to this environment and use that to extrapolate how we think it might be responding in different aspects of the human body. One of the aspects of microgravity that we think is influencing Candida albicans is low fluid shear. And if you'll go to the first slide, to try and demonstrate what does fluid shear really mean, if you think of a stone in a body of water, if that body of water happens to be a waterfall or a rushing river, there's going to be a lot of influence of the water flowing over, rushing over the stone to that stone. In contrast, and that's a high shear environment. In contrast, if you think about that stone in the bottom of a lake where there's very minimal shear, the influence of the water is going to be very different on the stone. We basically think about cells growing in liquid very similarly. If a cell is growing in an active liquid environment, it's going to experience a high shear impact on its cell membrane, and it's going to respond according to that pressure. If it's growing in a low shear environment, the response of that organism is going to be very different. If you'll go to the next chart, we can actually reproduce these different levels of shear in the laboratory. So in the top left panel, you'll see flasks of media. That's how we typically grow lots of organisms, including these yeast. And those are very rigorously shaking experiments, so that's an opportunity for high shear growth, if you will. And then on the right, really the only way that we can really get a microgravity and a low shear associated with microgravity is to send the bugs in space, which hopefully they're going to do tomorrow. So the hardware that we use, it's in that image. I also have an example here. This is hardware that was developed by Bioserve, and what it does, our experiments are contained within these tricked out test tubes, if you will, so that there are 3 chambers, and the plunger is depressed on that to allow a sequential mixing of the reagents in those 3 chambers. So on flight or on station, the plunger will be depressed to activate cell growth, and then the next plunger will be depressed to terminate growth. So all of that will happen on station before they come back at the end of the month. Low shear is, and I have alluded to it already, low shear is also probably an environment that the yeast might access in the human body. So for example, the nail bed is probably a location of low fluid shear, as are the brush borders and the kidney. So again, that's why we're looking at the response of the organism to this environment, because we think we can extrapolate what we find out there and bring it back and predict what might be happening in the human body. We're anticipating a lot of data from these experiments, and if you'll go to the next chart. One thing that we're clearly going to be doing a large molecular analysis. We want to know what genes are expressed, what molecular pathways are activated, and what molecular pathways are inactivated while in this microgravity environment. We're also going to be looking at a lot of physiological parameters. So what's illustrated here, just 2 different ways to look at the cells. Under high shear conditions, these yeast grow in the circular form, they look like balls up there. But when they're exposed to low shear, as we've done in the laboratory, we see an increase in the elongated filamentous form, which is associated, basically the combination of those 2 forms is associated with infectious organisms. The next slide, please. The next chart. Thank you. We'll also be looking at the ability of these organisms to form multicellular communities or biofilms. And again, this kind of structure is protective for the organisms when they encounter environmental stresses. So we'll be interested to see if there is an increase in the number of cells that actually make these communities. And the next chart, please. Something else that we found in our ground-based studies that was very interesting is on the right-hand side you'll see cells that are plated and they grow as a community. These are actually visual to the naked eye. They're pretty normal-looking, smooth colonies, but cells that were subjected to low shear make, on the left-hand panel, these very convoluted 3-dimensional structures. And so we'll be really anxious to see if we find cells that make those types of colonies when they're grown in the microgravity environment as well. Let's see where. So basically, the experiments. The other thing I was going to mention is we're also going to be looking at antimicrobial resistance. We think that's important because we are looking at an infectious agent. And so ultimately, the experiments that we're looking at are to assess the risk to the flight crew, but also to learn what we can about the molecular and physiological signaling that low shear environment and an overall microgravity environment produces in these cells, so that we can sort of take the next step in understanding what happens when these organisms are actually growing in special niches within the human body. Thanks.
Thank you, Sheila. Scott?
Excuse me.
Well, I'm here to talk about everybody's favorite topic, blood and urine samples. We're very excited. Sorry, we're very excited about the SpaceX launch tomorrow. We're more excited for SpaceX to come home because what we're expecting is 384 syringes of urine and 112 tubes of blood to come home in this SpaceX Dragon capsule. These samples will support two of the experiments that my team has on board the International Space Station. The first one is what we call nutrition, simply enough. And that's a study looking at a broad-based biochemical set of parameters in blood and urine to try to better understand nutritional status and related physiological symptoms, things like bone metabolism, oxidative stress, you name it. We also have an experiment that these samples support known as ProK, and this is a countermeasure study where we're actually looking at using modifying the diet to help minimize using the diet to help minimize bone changes during flight. Now, to talk briefly about the way these samples are collected, I think the first picture shows Aki Hashide after one of his blood collections. The blood collections are just like they are when you go to the clinic here on Earth. We use a standard needle, standard tubes, and you can see he's smiling there, so it can't be all that bad. On the other side, one of the most common questions folks get when from NASA when you go out and talk is how do people go to the bathroom? And while we do have a typical, what we call a waste collection system, or you might call it toilet, just like your toilet at home, it doesn't collect any samples. So when we want to collect samples for scientific reasons, we have to have a different way to do that. And what we do is we have what's called a urine collection device. And I brought one of these here that you can see. This is essentially a bag with this is the male version with a condom cuff on the one end. There is a different adapter for females. The crew member voids into the bag. Before flight, we put in a small amount of a volume marker, a chemical that goes in the bag. They void in the bag, mix it up, and then out the blue port in this end, they take one of these syringes and we'll fill this up with urine. And this is what comes home. And again, we're expecting just under 400 of these tubes of urine to come home. The other bag goes into another bag and then essentially goes into the trash. So while it may seem very strange to some folks, my typical line is that it may be urine to you, but it's gold to us, and that there's a lot of science that comes out of this. We've been doing these experiments for a number of years now. Actually, yesterday marked the 6th anniversary of our first blood collection on board the International Space Station. Mike Lopez-Alegria did that on Expedition 14, and we've published a number of our studies over the years. One of the more recent studies published last month in the Journal of Bone and Mineral Research, and what we showed there is that crew members that eat well, that have good vitamin D status, that exercise hard, can maintain their bone mineral density. And as I said, we published that last month. The image you're seeing there is the cover of the September Journal of Bone and Mineral Research. JBMR is considered the leading bone scientific journal out there, and we were honored to be selected not only to have our paper published there, but to make the cover as well. And while there are some nuances to the data that we found, that is, we've not yet entirely solved the bone problem, we have shown for the first time in 51 years of human spaceflight significant progress in maintaining bone mineral density. Again, there's some things that we still need to do in terms of understanding changes in bone strength. There's some things we need to do in terms of optimizing exercise, in terms of optimizing nutrition. But again, the fact that we're making progress in human subjects during spaceflight is very exciting stuff. And again, that's why we're very excited for just about 500 tubes of blood and urine to come home at the end of this month. I'll throw back to you, Mike.
Okay, thanks, Scott. Well, not only do we have members of the traditional media with us, but we are pleased to be joined by members of the NASA Social, who are using social media to report on the happenings around the SpaceX Commercial Resupply Services Mission 1. And we're going to take questions now. We'll start here with Marcia Dunn. Please state your name and affiliation and to whom you're addressing your question. Marcia?
Marcia Dunn, Associated Press for Dr. Smith. How old are these samples? I mean, I don't know if you've been stockpiling them ever since the shuttle stopped flying. I don't know if you brought any back in May. And how important? Is it